The tingling from too much pineapple is doing something very close to what venom does.
The main difference is not the molecule. It is the way in.
Things you've never seen now revealed.
This is the world in detail.
Here's a sentence that sounds like an exaggeration and is not. A great deal of the venom on this planet was never invented for killing. It is built out of proteins that already existed inside the animal, doing an old and thoroughly domestic job, breaking down food. Somewhere along the way, a copy of one of those proteins changed address. It ended up produced in a gland connected to a tooth, and instead of being swallowed, it began to be injected. Same tool, different route. If you want to feel the idea rather than just read it, eat too much fresh pineapple and wait. The tingling on your tongue is not the acidity. Pineapple carries an enzyme that breaks proteins apart, and the nearest available protein at that moment is the inside of your mouth. The jar of meat tenderizer in the kitchen usually contains the same class of thing.
Before the mechanism, it helps to know how ordinary this is. Venom is not some rare speciality of snakes. It appeared independently in a great many different groups of animals, which biologists call convergent evolution. There are venomous snakes, spiders, scorpions, jellyfish, several fish, marine snails that hunt with a chemical harpoon, centipedes, bees, and wasps with complex toxin mixtures, and even a mammal carrying a venomous spur. Each of those groups arrived there by its own route, at different points in time, solving broadly similar problems. Which is exactly why this subject matters. When nature finds the same answer many separate times, that is a strong signal the answer was close at hand, assembled out of parts the animal already had lying around.
The mechanism has a name and is surprisingly simple to tell. Genes duplicate by accident, and that is common. Instead of one copy of an instruction, the organism now has two. One copy carries on doing the original job and cannot drift far, because the animal depends on it. The other copy is free. It can accumulate changes without breaking anything, and it can end up being produced somewhere else in the body entirely. When that free copy starts being expressed in a gland attached to a bite, and when the changes it accumulates make the molecule more stable or more aggressive, natural selection begins favoring precisely those versions. This is why so many toxins are recognizable. They look like digestive enzymes, like clotting proteins, like molecules that regulate blood pressure. They are close relatives of things the body was already manufacturing. Venom is, to a large degree, repurposing.
And here comes the caution this episode cannot skip. This holds for a number of toxin families, not for all of them. There is genuine live scientific disagreement about how far the story generalizes. One line of research proposed that venom in reptiles has a single very ancient origin, in a common ancestor of a large group, which would make many animals now considered harmless carriers of traces of that system. Another line, revisiting the same data with different methods, argues that there were multiple independent origins and that the single origin hypothesis was pushed too far. Both readings are published, both carry serious evidence, and the argument remains open. So the honest sentence is this one. Many toxins came from repurposed body proteins. That part is well documented, and how many separate times that repurposing happened is precisely what is still being argued about.
But the strangest part of this has not happened yet, because this tool, which had already switched sides once, moving from digestion to hunting, switched sides a second time, and on that occasion, it was our species that changed its address. What began as a stomach enzyme and then became a weapon ended up inside a pharmacy box.
The best-known chapter of that third address begins in Brazil in the 1960s. A Brazilian researcher was investigating why the venom of a snake, common across the country, dropped the blood pressure of its prey so efficiently and identified molecules capable of amplifying a substance that widens blood vessels. That finding was one of the threads leading to the development of an entire class of drugs for high blood pressure. Today, among the most widely prescribed medicines in the world, and it is not an isolated case. There is a diabetes medicine developed from a molecule found in the saliva of a desert lizard, and a powerful painkiller derived from the toxin of a marine snail that hunts fish. It is worth saying plainly that this is pharmacological history, and nothing here is health guidance. The point of the episode is the route the molecule took, not the use of it. The same part was food, then a weapon, then a medicine.
It is worth leaving the laboratory to look at who lives alongside this, because that part rarely gets airtime. Snakebite is a documented public health problem, concentrated in rural areas of tropical countries, and it falls disproportionately on people who work the land, those who harvest, those who clear ground, those who walk through undergrowth early in the morning in rubber boots and long trousers. The anti-venom that treats it is an old and laborious technology. It involves extracting venom from animals kept at an institute, giving controlled doses to horses, collecting the plasma, and purifying the antibodies. Which means there are people whose daily work is extracting venom. There are horses in a field who are part of that chain. And there is a glass vial that has to be in the right place at the right time, at the end of a dirt road. It is a chain of people, animals, and distance, and it holds up the practical side of this subject.
It is tempting to cast the animal as the villain, and biology does not cooperate with that framing. Venom is expensive to produce. It takes energy and time to replace, and an animal that spends venom carelessly has none left when it needs to eat. So in most groups, the primary function is feeding rather than defense. There is even a documented phenomenon called the dry bite, in which the animal bites without injecting, because injecting would be a waste. Most incidents happen where the animal was stepped on, cornered, or handled in an encounter, not in a hunt. None of that makes an accident less serious or the risk less real, and none of it is an invitation to go near anything. It is simply the correct description of what is happening. This is not an animal attacking an enemy. It is an expensive tool being spent by a creature that would rather not spend it.
Where this leads is what makes the subject large. Every venomous species carries its own mixture, and those mixtures contain enormous numbers of distinct peptides with highly specific effects on ion channels, on clotting, on blood pressure. Specificity is exactly what pharmacology hunts for, because a molecule that hits one target tends to cause less unwanted effect elsewhere, which is why the world's venoms are often described as a molecular library, and it is a library, most of which has never been cataloged. There is a consequence of that which rarely enters conservation arguments. When a habitat disappears, a section of that library disappears with it, unread. In parallel, anti-venom production is beginning to change, with research into antibodies produced by engineering rather than extracted from animals, which would reduce dependence on that entire chain.
The honest counterpoint is that the story of venom as an infinite source of cures is considerably inflated. Decades of research have produced a small number of approved medicines derived from toxins. Not because the idea is poor, but because the road from an interesting molecule to a safe drug is long, expensive, and full of failure. Most promising compounds do not survive testing, so both sentences are true at once. Venom is among the richest sources of highly specific molecules nature has produced, and it has delivered few medicines relative to the size of the promise usually being sold. Neither statement contradicts the other. It simply puts the expectation where it belongs, which is what any genuinely interesting scientific subject deserves.
Which brings us back to the pineapple. Once you know that the same family of molecules can be tenderizing a piece of meat, dropping the blood pressure of a prey animal, and controlling the blood pressure of a person, something shifts in how the living world looks. Nature almost never invents from nothing. It takes what is already there and changes the address. A gene duplicates, the copy ends up in a different tissue, and a new function appears without anything having been created out of thin air. It is the same pattern behind nearly every good story in biology, and it is the pattern this channel exists for. Venom is frightening, and it deserves real respect. But it is not some monstrous exception to nature. It is an old, ordinary part being used somewhere new, and in the end, that is the one thing evolution does extremely well.
If your tongue has ever tingled from pineapple and you never knew why, now you do, and it is a good thing to bring up at dinner. Tell us in the comments whether you already knew about this link between digestion and venom. We read them all, and in the next episode, we stay with exactly this idea of a reused part. And look at something sitting right in front of you, the color blue. Almost no blue animal on Earth contains blue pigment. The butterfly, the feather, the beetle that shines. Nearly all of them use a completely different trick to produce the same color. And once you know which trick it is, you cannot look at anything blue the same way again. If you like finding the mechanism behind the obvious, subscribe.